My name is Xiangzhu. Today I'm talking about VCP protect against the proteoperoxide seedings. So first of all, what is a VCP? VCP is an ATP nest that take a lot of functions within the cells, and it also cause VCP mutations also cause a disease called multisystem proteoperoxidase (MSP). So it can affect many different tissue types, including muscle, brains, or the bones. One of the common features of VCP patients that they all have proteopathic inclusions, including TTP43, as they demonstrate by neurons over here. And recently they found that it can cause alpha-synuclein inclusions or tau inclusions in the brain as well. So the questions in our lab ask is, what is the relationship between VCP and the proteopathic proteins? First of all, we need to know what is the proteopathic proteins. It first recognized as a biomarkers in many neurodegenerative disease, including AD, Parkinson's, frontotemporal dementia, ALS, and etc. They share a common features of unstructured domains, which is really liked to form aggregates. Also, they are kind of like infectious. They can spread from one domains to one regions to another's, and using the already formed aggregates as a template to form a new one. In general, those proteins are toxic to the cells, and the accumulations of those proteins is highly correlated with disease progressions. So the spreading, we call the spreading of those proteins as the seeding events. So we use the FRET biosensor line called alpha-synuclein CFP-YFP sensor lines. It will generate the FRET signals when the alpha-synuclein aggregates to monitor the aggregations events. Using these platforms, we generate a library containing more than 90,000 different genes and sort out the gene factors that affect alpha-synuclein seedings. And we get the plots over here. We have some of the heats that the red dots are the heats we think that protect the genes that protect against the alpha-synuclein seedings. And within the secondary validations, we find the VCP is the one that gives us the biggest change out of the selected hits we validated. So we turn our focus on the VCP. First of all, we validated into another system the primary neuron cultures, and we find that when we knock down VCP in the primary neurons, the alpha-synuclein is more likely to make aggregates, as they show over here, which is phenocopy what we find in the alpha-synuclein biosensor line. Also, we use VCP inhibitors, which are in a reversible way, temporarily inhibits VCP functions. And we can, you can see over here, the ML240, one of the VCP inhibitors, actually get a similar effect as a VCP knockdown. Then we ask if VCP disease mutations also have effects on proteoperoxide seedings. As we know that VCP disease mutations specifically affects part of the VCP functions without affects or decrease the VCP ATPase activities. And we can see in the alpha-synuclein cells with the cells that overexpress VCP different vectors, VCP disease mutations vectors actually significantly increase alpha-synuclein seedings compared with the wild-type overexpressed cells. And this effect is also seen in the hippocampus neuron culture, which have VCP R155 mutations. Finally, we want to see the effect, the VCP mutations effects in vivo. We inject alpha-synuclein seeds into the skeletons of the VCP mutations mice over the over the 67 mice, and we find that similar to what we've seen in the different cells models, VCP disease, two different VCP disease mutations mouse model generates more alpha-synuclein aggregates compared with the 67 controls in different areas, including cortexes. Then we ask if VCP effects on the proteopathic seeding is alpha-synuclein specific, or it happens in the proteopathic seeding in general. So we use another different proteopathic proteins called TDP43, which is commonly found in VCP disease VCP patients, and we can find both VCP inhibitions or VCP disease overexpression can increase TDP43 seeding, similar as alpha-synuclein seedings. These effects also found in tau seeding as well. The data is now showing over here. Also, we repeat these results in the TDP43 seedings in hippocampus neuron culture, and we find the similar effects over here. You can see VCP disease mutations have more aggregates, generate more aggregates compared with the wild-type control. So in summary, we use CRISPR-Cas9 whole genome library and discovers genes involved alpha-synuclein seeding, including VCP. And VCP affect proteopathic seeding both in vitro and in vivo. First, VCP inhibitions aggravates alpha-synuclein seedings, both in cell models and on mice. Also, VCP mutations increase TDP43 seeding and tau seedings. It suggests that VCP is a shared pathways for proteopathic seeding in general. So since our lab, our collaborator, my lab mates, and the funding that support this study. Thank you so much, and thank you for listening.